protein synthesis. What is DNA and what does it do? DNA is deoxyribose nucleic acid. It essentially carries the genetic code in a double helix structure, as I'm trying to demonstrate over there. The components of DNA are adenine, thymine, guanine, and cytosine, all of which amino acids and nucleotides. The way they pair is A is equal to T and C is equal to G. These are all nucleotides that make up the nucleic acid of DNA. All of these nucleotides are composed of a 5-carbon sugar called deoxyribose, a phosphate group, and a nitrogenous base. It's called deoxyribose nucleic acid because the DNA is made of nucleotides, which are made of deoxyribose. All of the nucleotides in one strand of DNA are held together by covalent bonds. It is possible for many nucleotides to be arranged in any order, and that means with many different combinations they may function differently. What is DNA replication? DNA replication is when DNA is basically duplicated during mitosis or meiosis. It is carried out by a group of enzymes. First, they open a molecule of DNA by disrupting the hydrogen bonds between the base pairs. Unwinding the strands, each strand then becomes a template for attaching complementary bases. The main enzyme facilitating DNA replication is called DNA polymerase. While producing the sugar phosphate bonds that join nucleotides, this enzyme also ensures that each DNA strand is a near perfect copy of the original. This is an example of how the DNA polymerase comes in, and then they get joined, and then they become a new form over there.
What is this?
That's helicase.
What's helicase?
That helps them bond.
Okay.
Helps them bond. What is RNA and what does it do? RNA, like DNA, consists of a long chain of nucleotides that consists of 5-carbon sugar, phosphate group, and a nitrogenous base. But RNA differs from DNA in 3 ways. 1. The sugar in RNA is ribose, deoxyribose. And 2. RNA generally is single-stranded and not double-stranded. RNA contains uracil and not thymine as one of the bases. RNA may function, but its most important function is protein synthesis. RNA manages the assembly of amino acids into proteins.
That word has many functions.
Okay.
You will fix it?
Okay.
Okay.
So this diagram is showing how what are the different types of nucleobases, and this is the structure of DNA, and this is the structure of RNA. If we see uracil is here in RNA, and thymine is here in DNA. And so C will pair, cytosine will pair with guanine, the adenine will pair with thymine, as you see, they pair here and they pair here. And for uracil and adenine, they're right next to each other here. Then cytosine and guanine still pair together.
Blue and green? No?
Huh?
Blue and green are not pairing with each other.
I don't know how.
You said cytosine is and
Yeah.
no, but here in RNA. Okay.
See? Like this?
Okay.
Like this.
Oh, okay. These two and these two. Gotcha.
Transcription, and what is the purpose of it? Most work of making RNA takes place during the transcription process. Segments of DNA serve as templates to produce complementary RNA molecules. Transcription requires an enzyme called RNA polymerase, that is similar to DNA polymerase. RNA polymerase binds the DNA during transcription and unzips the DNA strands using one strand of the DNA to assemble the nucleotide into complementary strand of RNA. Copying a single DNA sequence into RNA makes it possible for a single gene to produce hundreds of RNA molecules. All of this takes place in the cytoplasm.
And what is this doing?
This is cytoplasm.
Okay. And what's inside this? What's this?
RNA and DNA.
Okay. And what's this? One
What?
single bit.
That's the nucleus.
Thanks.
And this is kind of demonstrating how it's taking the key components and it's building its separate strand of DNA on the bottom.
Cool.
And then RNA polymerase is taking it out. Translation of genetic code in the cell. Four different bases in RNA make the total number of 3 base codons in the genetic code 64. Most amino acids can be specified by more than one codon: UUA, UUG, CCU, CUC, and CUA, and CUG. Specified leucine, but only one codon of UGG specifies the amino acid tryptophan. Genetic code also has a punctuation mark to start or stop codons. The codon AUG methionine is a start codon for protein synthesis. Following this codon, our mRNA is read 3 bases at a time until it reaches a stop codon, which ends the translation and completes the polypeptide. This sequence of bases in the mRNA molecule is a set of instructions which is the order in which amino acids should be joined to produce a polypeptide. Completed polypeptides fold into shape or joined with other polypeptides to make a functional protein. Ribosomes decode the sequence of codons in mRNA to assemble amino acids into polypeptide chains. This process is known as translation.
Yes, definitely.
So you see it'll go from RNA to a protein then to DNA. And it starts from RNA, from DNA, then it goes to transcription. Then you have the different types of RNA, messenger RNA, ribosomal RNA, and transferred RNA. And they'll go to the ribosome, then the translation happens and you get proteins. The importance of these processes. DNA maintains the genetic information in the nucleus, and RNA takes that information into the cytoplasm where specific proteins are constructed. The mechanism is very easy in prokaryotes. In eukaryotes, transcription factors help express a particular gene in a particular cell type. This matters because cells throughout the body are specialized and express different sets of proteins: keratin in skin and hemoglobin in the marrow of the bone. And mRNAs enable the cell to express only the genes it wants, not the entire DNA genome. Mutations. Error in the transcription or translation process can have a change in the base sequence and is called a mutation. It can have the following effects: no change in the phenotype. Some mutations do not have any noticeable effect on the phenotype of an organism. Small change in a phenotype. A single mutation caused a cat's ears to curl backwards slightly. Another one would be different colored eyes. Big change in phenotype. Some really important phenotypic changes like DDT resistance in insects are sometimes caused by single mutations or mutation that causes sickle cell anemia. importance of molecular biology. The study of DNA and RNA molecules is called molecular biology. Advances in technology have resulted in the mapping of the entire human genome. It has improved our understanding of several diseases like HIV. It has implications in developing solutions and treatments for several diseases like Alzheimer's disease, Parkinson's, and many more. Growing tissue from stem cells is used for treating diabetes and any other treat solutions for several diseases.